Welcome to Going Antiviral, the podcast for the IAS-USA, a professional continuing medical education organization focused on HIV and other viral diseases. I'm Dr. Michael Saag, professor of medicine and infectious diseases at the University of Alabama at Birmingham and volunteer member of the IAS-USA Board of Directors. Welcome to the Going Antiviral podcast. Today, March 31st, 2025, we are speaking to Dr. Raymond Schinazi, who holds several positions at Emory University School of Medicine, including the Francis Winship Walters Professor of Pediatrics and is Director of the Division of Laboratory and Biochemical Pharmacology. He leads a research group with expertise spanning medicinal chemistry, virology, molecular biology, toxicology, pharmacology, immunology, and computational biology. Dr. Schinazi has 5 honorary medical degrees and is a Knight of the French Legion of Honor. John Cohen from the journal Science called him the King of Pills, owing to his development of so many antiviral drugs. Today, Dr. Schinazi joins us to discuss his role in developing the cure for hepatitis C. Ray, welcome to the podcast. It's great to have you.
Hi, Mike. Nice to see you again.
It's wonderful. So let's just start at the beginning. Tell us a little bit about yourself, your background, and how you ended up in Atlanta.
Well, basically, I moved from 3 different continents. I started off in Egypt, where I was born. Due to political situation, I had to move to Europe and end up at boarding school, and then went and did my studies at Bath University, where I studied chemistry, both a bachelor's and PhD. My dream was to come to the US, so I got an offer from William Prusoff at Yale University to study antiviral agents. Of course, I was quite familiar about, I was very familiar about herpes viruses because my uncle, Andy Namias, was a herpes expert for many years, and I knew about herpes even when I was 12 years old, so I managed not to get herpes. So that's a good thing. But on the other hand, I learned a lot from both Andy and from Bill Prusoff. At the same time, I had the opportunity to train with Bernard Roisman at University of Chicago, and at the time with Yung-Chi Chang, Tommy Chang at UNC, University of North Carolina. So I had really a lot of postdoctoral fellowship, a lot of opportunities to learn the whole process from chemistry to biology. And when I moved to Emory, I stopped being a chemist and I started being a biologist and a virologist. And that's how I came along. And then, of course, HIV occurred. And then after HIV, there was HBV. And then from HBV, we went on to HCV.
Yeah. So tell us a little bit. I know you were heavily involved with development of antiretroviral agents for HIV. Just briefly about that, because I really want to dig into the Hep C story.
Well, we started off with D4T with Bill Prusoff, and that was a successful story because it was a much better drug than AZT, but they still had deficiencies. And then working with Dennis Liotta at Emory University, we were successful at making oxyethylene nucleosides, which are basically the backbone for what we know today as emtricitabine and lamivudine. These compounds have really one of the most successful nucleoside analogs on the planet because they are used in many cocktails even today, and they've made a big difference and saved millions of lives. So I'm very proud to have worked with Dennis and, of course, the pharmaceutical industry to bring this to the patients. And I think we've done an amazing job with these molecules, which are completely novel at the time because nobody imagined that L-nucleosides could have antiviral activity. And we were the very first people to establish that and made, of course, FTC and lamivudine.
So you took FTC to, is it Triangle Pharmaceutical? Is that right?
Oh, it's a whole story because it went from Emory to GSK, what is GSK today, and then when GSK had acquired lamivudine, which was also one of our drugs. Basically, and there was a lot of controversy about it. So I don't want to go into legal issues because whatever that's been resolved. And then Triangle, people from GSK or the Glaxo part of GSK formed Triangle Pharmaceutical with me and a couple of other scientists, and we developed FTC until Gilead took it over.
Right. And that link to Gilead will come back later in the story. So you have this successful run with HIV. Where did your interest spark about hepatitis C? What was your vision as that project emerged out of your lab at Emory?
Well, it was at the time, of course, we thought we had the best-in-class nucleoside analogs for both HIV and HBV. Remember, lamivudine was the very first oral drug for hepatitis B. And we also had telbivudine, also I'm the sole inventor of telbivudine. That's another HBV drug. So we had done enough on HBV. Of course, we hadn't cured it. We hadn't cured HIV either. But we thought, what else could we do? And the timing was perfect because real-time PCR became available, and the replicon system from both Ralph Battischlager, and Charlie Rice became available. So that really, we decided to invest in that, and we formed a company called Pharmacet. And we'd done some work. We tried to get funding for HCV, but everybody thought that HCV could be cured with immunology. Everybody. And we couldn't get any funding, hardly any funding. There was a lady at NIH called Leslie Johnson, you may remember her, and we just couldn't get anything from her. She was all spending all the money on immunology. Well, we proved them wrong. And in fact, we now have a wonderful molecule that actually cures, not just treats, but cures. It's not a band-aid. It's something that makes a huge difference. So basically, having the replicon system and having terrific scientists at Pharmacet, people understood the genetics of the virus, and had assays using real-time PCR rather than Western blot, we were able to screen some of our library of compounds that we had, and we quickly discovered that a compound called 2'-fluorocytidine was effective. And from there, it evolved to what you would know as gemcitabine, which is a compound used for cancer. Of course, you can't really use it as an antiviral, but it does have antiviral activity as well, although it's toxic. And then we came up with the idea of coming up with a methyl fluoro, and the fluoro being very, very important at the 2' position, because that actually reduces mitochondrial toxicity. Nobody in his right mind would put a fluoro based on the studies that have been done with FMAU, but we had the idea, and we put the fluorine at the 2' position in the down, not the up. Up would be maybe more dangerous, but having it down and have the methyl up, that really made a difference. And many other drugs were made with a hydroxy group, and they all failed, and in some cases killed 4 or 5 patients. As you know, tragically, this happened with the Inhibitex compound. So it's very sad that this happened, and we knew there was toxicity with that compound. Unfortunately, people didn't pay attention. But clearly, the drug that we have, the sofosbuvir, was really a tremendous breakthrough, a real magic bullet, a compound that made a huge difference because of its safety, not just its activity and its potency, but safety and the fact that it got degraded in the liver specifically using the prodrug system. So the discovery was a bit serendipitous because we were working on a compound called PSI 6130, which is a cytidine analog. And we discovered I should say, analytical chemist in our group with biochemical experience, Morikami. He now works at Gilead Pharmaceutical, but basically he worked for us. He's originally from Karen Anderson's lab, and he discovered that the C analog gets deaminated at the monophosphate level, and the monophosphate is then phosphorylated to the triphosphate, and the triphosphate is effective. So it didn't take much of a genius to figure out that if you make a prodrug of that molecule, you will get delivery of the U analog intracellularly as a monophosphate, and that would then lead to what we know today as sofosbuvir.
Yeah, so this is all around what, 2004-ish?
Approximately, yes.
Yeah.
Yes.
And
2004, 2005. Actually, the original papers were published in 2005, and some additional papers came out afterwards from our group and from Pharmacet, and then of course Mike Sophia with his paper based on the studies that were done at Pharmacet. He was actually an employee of Pharmacet and basically made the prodrug, but that was pretty obvious. The part that's not obvious is how to make the warhead. The warhead is the key,
yeah.
not the prodrug. We knew already that we knew a guy called Jeremy Clark. I should give him credit. He doesn't get any credit, but he is the chemist who actually made the precursor, the U and the C analog of what we call sofosbuvir. He didn't add a prodrug. We had tried some other prodrugs, but not the McGuigan type of prodrugs, but they are in the patents, the original patents. And he's the sole inventor of this molecule. He was an employee of Pharmacet at the time.
Well, and we now know. I mean, just for everybody who's maybe not familiar, hepatitis C first really discovered in '89. Interferon and the immune therapy, as you were talking about, became all the rage, especially among the GI docs who uh, really focused on the immune-based responses. Then you come in with your experience with nucleotides, and come up with sofosbuvir. But that by itself isn't going to be enough, right? You need an NS5A inhibitor. So how did you end up uh,
Yeah.
figuring out
Well,
that?
you remember at the beginning we use we sofosbuvir itself. If you treat long enough, you can actually cure everybody just with one monotherapy. You don't need second drug. But we want to shorten treatment to 12 weeks or even 8 weeks. So they use interferon at first. That was already on the shelf, already available. Both Sharing and Sharing Plow was making it, and so was Roche at the time. And they basically, it was working very well. But of course, you had the injectable, and you didn't need to treat for a whole year. You only treated for as long as you treated sofosbuvir, and you're getting very good responses. But nobody wanted to take injections except in certain countries. So having the discovery of NS5A inhibitor, especially with daclasavir, which is the compound made by BMS, it's a superb molecule. Beautiful story. You should make a story about the discovery of this molecule as well. It's really a fantastic story, and how they came about it, and it was pangenotypic. So the idea was, ideally we wanted to combine daclasavir with sofosbuvir. That would have been a great formulation, but unfortunately the companies couldn't get together. But in Egypt, by the way, the generic form of sofosbuvir is they use DAC plus sof. So that's available in Africa today. But you know, Gilead developed its own NS5A inhibitor, and that took time. So they had to partner with other people initially, but they didn't make a single pill like they've done for HIV. But eventually they developed their own drug, and but they wasted a few years, in my opinion. Could have been done a lot quicker if they had partnered with BMS.
Yeah. And did Pharmacet ever develop an NS5A inhibitor, or is that just came from
Yes,
Gilead?
it did. It did have, but by the time the others were already on the market, by the time we had actually better compounds, and we had nominated the compound at the time, but we never pushed it. There are some patents out there. You can look it up.
Yeah,
So we
yeah.
did work on NS5A.
Okay. So you develop a kind of a stable of drugs that can work for Hep C, and you're trying to get um, this the product picked up by large pharma, in particular Gilead, I suspect. Um, what was that like? I mean, a lot of people
Okay.
don't think about it from a pharma perspective. You think about the medicinal chemistry, but take us inside your world from
Yeah.
2005 and six onward.
So
Yeah,
the business side of drugs, which is fascinating actually, because not many people in science get the opportunity. But we developed a strategy, and the key person was a person by the name of Abel de la Rosa, who I hired as a business development person, and he was smart. I'll give him full credit. He basically approached every single pharmaceutical company that had a potential candidate for HCV, or and actually offered them sofosbuvir free of charge to do their own clinical trials. So he had about 5 or 6 companies, from Boehringer Ingelheim to Bristol to Gilead and others, Merck and so on, and they all came up with great results. So they all had the results, but weren't privileged to these results. So we gave them the drug for free, and they put it together with their molecule, and they got fabulous results, all of them. And that's where the bidding war started, and that's how we ended up with $11.4 billion for this molecule. At the time when we started, the company was worth maybe about $3 billion. So that was really exciting. And I raised my hat to Abel de la Rosa, who did a fabulous job.
yeah. And so that ultimately went to Gilead, and they paired it with ledepasvir. Is that right? And that became
Correct.
the trade name
They, they
Harvoni.
did they did interferons first because they had no
Yeah.
other drugs, but then finally they got their own NS5A.
Yeah, I remember vividly around 2010, maybe 2011. Then the studies are starting to come out with all oral therapy, 12 weeks, um, working in the 90%, 90% plus cure, maybe 95, 98%. And I remember I was at the GI meeting when they presented those first results. And again, the orientation was, yeah, these antivirals may help, but you're still going to need a kick to the immune system with interferon. And the presentation went on and it showed this incredible response. And there was stunned silence in the room. And it was just a remarkable time because usually the hepatology meetings are very vivacious and a lot of conversation, and this was just silence.
Yeah.
And
I
it's
actually
really,
was in. I was trying to get into the room and I couldn't.
yeah, that's ironic, huh?
So I was at the door trying to listen. It was packed room at the time, and it was quite exciting. So we actually had a meeting. The pharmacist group had a meeting, and we named the drug actually between among ourselves as Perfectovir, the perfect drug. And we because we knew the results and we knew it was very successful. But remember that Gilead did spend time giving soft with interferon, and for a long time, so they had experience with that, and they knew it was not going to be perfect. So having the NS5A inhibitors make a big difference. And in this particular case, we got lucky. We didn't need the immune system at all.
Yeah,
The drug was so powerful. Of course, the problem with the drug is that you get reinfected. So hopefully people who take these drugs don't get reinfected. And that's the key. And that's why we also eventually we'll need a vaccine for hepatitis C, and people are working on that. But in the meantime, I think people should be careful with using this drug. And once they use it, they should try to stay safe and not to get reinfected.
yeah. I think in the Amsterdam group showed among their cohort of men that the rate of reinfection was up to 3% or so over the course of a couple of years. So
Yeah.
it is out there. But maybe if we get enough people treated, enough people cured, then that risk would, by nature,
Yeah.
just kind of go down.
Yeah, I just like to add one more thing. I mean, remember the basic science. The U analog of what we know today as sofosbuvir, the parent nucleoside, is totally inert and totally inactive. If we hadn't done the biochemistry, the studies on the pharmacology and biochemistry of this compound, we would never have moved that compound forward.
Yeah,
It would be, and now, of course, when you have a U analog as a chemist, as a biologist, as a virologist, you actually make the prodrugs immediately and test them. But at the time, it was not obvious at all. At all, you would throw it away in the garbage of failures. So this was really a truly innovative discovery,
yeah,
and people need to appreciate that it wasn't just an accident. It was really beautiful work done. We had already shown that the compound, the 6130 I mentioned earlier, was deaminated in monkeys. And, we were disappointed with that because if it gets deaminated, it's not going to be active. But guess what? If you make a prodrug of the deaminated products, you have sofosbuvir. So it's a amazing discovery.
yeah. And I'd also like to let's go back to when you created the compound and when you first had an inkling that this could be a huge hit. What was Tell us about that and what that was like for you personally.
Yes, we only made about 3 milligrams of compound. We didn't have much. We may test it once because we had the systems working, very robust system, and the systems really didn't hardly never failed. We actually had, believe it or not, we had NHC as a positive control. NHC is the compound monopiravir-based molnupiravir. We discovered also that compound, by the way, was discovered in Pharmacet as an antiviral, but we never developed it because it had mutagenic effects, and it's still we didn't want to touch it because it had mutagenic effect, but it was an interesting compound. But that was used as a positive control because at the time nobody had a positive control. So we had the positive control, we had the assay, we had the replicon system, we had the multiplex system. So the PCR, real-time PCR with multiplex, so we can control not only the antiviral effect but also the toxicity of the compound simultaneously. And we knew the compound was essentially non-toxic. Of course, we went through a whole battery of compounds of tests, including mitochondrial toxicity, peripheral neuropathy, and so on, to reduce the risk as we go towards the clinic. And then, of course, the toxicological study. But remember, I knew it was a U analog, and I knew the U analog when sofosbuvir is degraded in the body, it will form U, and I know the U analog is totally inert. which doesn't always happen with the other nucleosides.
Yeah.
So I knew it was going to be a very safe drug, and it was. It is a very safe drug, relatively speaking. I don't know of any major side effects or any black box or anything about this compound. It's
Mhm.
actually very clean. Like FTC. FTC is
Yeah.
also very clean.
Yeah.
Usually we go through the whole battery of tests. So I was very excited, and I was immediately talked to our lawyers and filed a patent. It was very competitive at the time, and I didn't know if anybody else had come up with the same idea. But we immediately went and filed a patent, and of course added value to the compound by doing additional testing. It wasn't easy to make this compound. Many chemists at Pharmaset had tried and failed. I led the chemistry group at Pharmaset. I not only was I basically the executive board chief, the board of I was on the board of director of the company. I ran the company, but I also ran. My passion was running the chemistry group, and we had tried. Many chemists in our group had tried to make this compound. It was not trivial. So by learning how to make the sugar part with the fluorine and metal, we were able then to couple it and make about 3 milligrams. But later on, we made 3 kilos of the compound, and we had to refine the process to make it. And it cost us millions of dollars to make, just by the way. People don't talk about that, but it's very expensive at the time. Of course, today it's much less expensive, but at the time, it was basically more expensive than the most precious metals around,
Hmm.
at the time. So we had very limited supply, but then we made 3 kilos. So that's the whole process team at Pharmaset developed a great, and it involved a Korean company, a Brazilian company, and our group working together. Really international effort to make the 3 kilograms of material.
Yeah. And then you just went off the replicon and the multiplex to determine its potency. To
Yeah,
show
that was the key.
yeah.
That was the key. We timed it right. We didn't have much money, but in those days, these machines were expensive, and we were a startup company.
Yeah.
And we made the investment, and I think we had great scientists who convinced me to buy this equipment. You're a bit stingy at first when you have a startup company, and you only had maybe $3 million in the bank, $3.7 million in the bank at the time. So you need to decide how you're going to spend your money, and the lawyers obviously take the bulk of the money. That's one of the problems, as you well know, in forms companies. But
Yeah.
and the patents and the legal issues are enormous. But we were able to manage that, and it worked out well. Had a very tight team, excellent scientists, excellent chemists, excellent biologists.
And there's no animal model per se that is for Hep C. Not really.
At the time, there was only the chimp, but nobody could afford the chimps. There was a
Yeah.
group at Southwest in San Antonio, Texas, that had a chimp model, but eventually they banned the use of chimps.
Yeah. So that means that you have to do your first studies in humans and look at sort of reduction in viral load, I guess similar to what we did in
Yeah,
HIV.
yeah. There were a lot of animal models available at the time. They were not perfect. They hadn't been proven because nothing really worked well in these models. There was in Canada and other places models in humanized mice, but nothing really was available. So all you have to do is do the toxicology. The idea was we have a compound, we know the mechanism of action, which is basically inhibition of the HCV polymerase. We knew it was specific. We knew it was non-toxic. We had a lot of toxicology data, and you go off to the FDA, talk to them, and they agreed. I mean, there are other compounds. Many compounds have been tried on HCV, as you know, and failed.
Yeah.
There's a whole cemetery full of dead drugs that failed. And I think it was in Michael Fried. If I don't know if you know Mike, but
Sure.
Mike Fried actually has published more papers on negative results on HCV than anybody else I know. He worked with Jay Hoofnagel at the time at
Yeah.
NIH.
Yeah.
So he was a consultant actually for Pharmaset and helped us, and I'm very grateful to him.
Uh,
So
I wonder
he
if he
was.
was. I wonder
Yeah.
if he was one of the fellows during the FIAU story that you referenced
I
earlier.
don't know. He never admitted to me about that.
I
I
know
know
that was
Jay
that,
was involved, of course, but they were all very weary about nucleosides. But there are
sure,
some great
especially
there
with
are
the
some great
fluoride,
nucleosides.
especially with
Pe-
that fluoride group you were talking about.
Yeah, yeah. People forget that acyclovir is a nucleoside. Pencyclovir is a nucleoside, and famciclovir is a nucleoside, and FTC is a nucleoside, 3TC is a nucleoside, and sofosbuvir is a nucleoside. I mean, you got to get the right ones, guys. You need to have the proper testing. Come and talk to me.
Yeah. So what are you working on now?
We have an HBV drug that's moving along to phase 2 clinical trials. We licensed that to a company in San Francisco area called Aligos, and it's moving along in phase 2. It's a very interesting compound, super potent. At 10 milligrams, you get about 6 log drop. I wish we could get an HIV drug that can give you 6 log drop at 10 milligrams, but we don't have that. Maybe one day we will. So we bring down the virus load. It has multiple mechanisms. That's how you're able to bring the virus load to. And now it's being used at high dose at 300 milligrams and it's safe. You want to bring down not only the first phase, but also the second phase of the virus. And it's one of the few compounds that I'm aware of that can seroconvert people from HBe antigen positive to HBe negative. Yeah. So it does it very reliably, which is great. Tells you there's no virus present. And HBe is actually a marker for cccDNA. So you know that the cccDNA reservoir is coming down. What the impact is going to be long term, they've already dosed patients up to 92 weeks, which is a long time. They keep the virus suppressed. There's been no resistance being selected because, as you know, dead viruses don't mutate. And basically, they are now thinking, interestingly, to add interferon in combination with this compound called Aligos 184, the same principle that initially as we thought with HCV, to basically shorten the duration of treatment. I should also mention that HBs antigen comes down, HBCR, the core antigen comes down, but it hasn't gone down to undetectable levels. Yeah. Now in mice are not humans, but mice, a study by Hélène Strick-Marchand in a Pasteur Institute in collaboration with our group, which will be published shortly, demonstrates that you can get cures in mice with this drug, or with a cousin of this drug, I should say, a very, very much related compound called GLP-26. So we're very excited that we can actually do it in mice, and we know, the interesting is, we know why only half the animals became seronegative and seroconverted, which is very important. In the case of Aligos, we have not seen any patient seroconvert yet, but I think with interferon, we hope to see seroconversion much faster.
Yeah, interferon doesn't seem to want to go away completely, except if you've ever taken it, you probably want it to go away.
Yes, I know, but if we can cure hepatitis B, it's a beginning, it's a good start, because we don't have any markers for cure. The only ones people think is HBs antigen or this or that or the other. But if we are able to find what the markers are, we'll know when to stop therapy.
Yeah. Well, as I sort of predicted or thought, this time has gone by very quickly. Ray, thank you so much for being with us, but thank you even more so for all the work you've done over the last several decades to improve our approach to therapeutics for all sorts of viral diseases. And I know that you're very proud of that work, but it's made a huge impact on disease around the world. So thanks.
Yes, thank you, Mike. We're still going strong and we hope to have some additional news in the near future.
Great.
Thank you.
Thank you. Thank you for listening to this episode of Going Antiviral. Catch up on earlier episodes wherever you listen to podcasts, and check out the video versions of this episode and others on the IAS-USA YouTube channel. You can find these links in the show notes, or simply go to YouTube and search for IAS-USA, and there they are. Be sure to subscribe, rate, and review Going Antiviral on Apple Podcasts. This podcast is for informational and educational purposes only and is not intended to serve as medical advice. Opinions expressed in this podcast do not necessarily reflect the opinions of the IAS-USA or its affiliates. If you have a question you'd like answered on the show, you can send it to podcast@iasusa.org to be answered in a future episode.